Welcome to this audio slide presentation. My name is Birgit Waltenberger. I am from the University of Innsbruck and I will introduce you to our recent review about natural product agonists of PPAR-gamma, which was published in Biochemical Pharmacology. Metabolic disorders are of great significance today. The features of the metabolic syndrome include abdominal obesity, hypertension, dyslipidemia, insulin resistance, and glucose intolerance, as well as establishment of pro-thrombotic and pro-inflammatory states. People affected by the metabolic syndrome have a greater risk of developing cardiovascular diseases and type 2 diabetes. At present, the metabolic syndrome is already affecting more than a quarter of the world's adult population, and it contributes to mortality and morbidity worldwide. The two main approaches in metabolic syndrome management are lifestyle modifications and pharmaceutical interventions. For the treatment of patients suffering from type 2 diabetes, aside from lifestyle alterations, insulin and insulin analogs and a number of oral anti-hyperglycemic pharmaceuticals are used. Among others, these drugs include thiazolidinediones, which are agonists of PPAR-gamma. This figure shows the PPAR-gamma transcriptional activation. First, activating ligands bind to PPAR-gamma and to its dimer partner RXR. Then, heterodimers of PPAR-gamma and RXR are formed, which bind to the promoter regions of the respective target genes. Cofactors are recruited and the expression of PPAR-gamma target genes is initiated. PPAR-gamma is the master regulator of adipocyte differentiation, plays an important role in lipid metabolism and glucose homeostasis, modulates metabolism and inflammation in immune cells, and controls cell proliferation. Therefore, PPAR-gamma represents an interesting pharmacological target, which is able to simultaneously modulate several of the underlying pathologies of the metabolic syndrome. Endogenous ligands of PPAR-gamma act as weak agonists compared to the synthetic thiazolidinediones, which are applied in type 2 diabetes. In spite of being effective in normalization of blood glucose levels, these full PPAR-gamma agonists have serious side effects, which makes the discovery of novel ligands highly relevant. The PPAR-gamma activating potential of a wide range of natural products has been explored. Here you can see some of the most interesting examples of investigated sources. Noteworthy, PPAR-gamma ligands were often identified in plants that are common food sources or used as traditional medicines. A selection of natural products, well characterized as PPAR-gamma ligands, is presented in this review. The majority of identified natural compounds are rather weak agonists of PPAR-gamma, often activating the receptor as partial agonists. In vivo studies suggest that some of these natural products improve metabolic parameters in diabetic animal models, partly with reduced side effects in comparison to thiazolidinedione agonists. Depicted in this figure are the binding modes of selected PPAR-gamma ligands. The natural product amyfrutin B is a partial PPAR-gamma agonist, which binds to PPAR-gamma in a way distinct from the thiazolidinedione agonists. The neolignan magnolol is a dual agonist of PPAR-gamma and its dimer activation partner RXR. Similar to some endogenous ligands, it binds to PPAR-gamma as a homodimer. And the flavonoid luteolin binds to PPAR-gamma simultaneously with the fatty acid myristic acid. In conclusion, natural products prove to be a rich source for PPAR-gamma ligands. The majority of identified natural compounds are rather weak agonists of PPAR-gamma, often activating their receptor as partial agonists with activation patterns similar to endogenous ligands. Several PPAR-gamma agonists were identified from traditionally used medicinal plants, culinary herbs, beverages, or food sources. The identified PPAR-gamma activating natural products and plant extracts bear a good potential to be further explored for therapeutic effectiveness, as well as to be studied as potential dietary supplements to counteract the metabolic syndrome and type 2 diabetes. For further information, please find our review in Biochemical Pharmacology. Thanks for listening.